So thank you, Nathan, and the other organizers for having me in this wonderful meeting. It's such a privilege being after the Mother of the Year. Okay, so my lab is interested in molecular mechanisms of neurogenesis and neurodegeneration, and we are most interested in those signaling systems that has components that are human-specific, that is, that only humans have them, and that potentially have contributed to the evolution of the human brain with all its special capabilities, but also special vulnerabilities. Today, I'm going to show you two research stories done in my lab. One is of the axon guidance system SLEET robo, and the other is of the neurodegeneration executioner protein SARM1. So starting with the SLEET robo. So SLEET robo plays a central role in axon guidance, which is the process by which neurons extend long processes. In giraffe, it can reach 3 meters. And these processes have to go through dense material in order to form the right connections. And in order to do so, they have to take the right turns and avoid taking the wrong turns. And SLEET robo plays a central role in that. And here you see live recording of one such axon that presents on its leading edge the growth cone roboreceptors that responds to SLEET bath application. And you can see how it repels it. So in essence, SLEET robo are, in the context of axon guidance, plays a role of a repulsion system. So based on that, we can come up with a basic model for robo activation in which robo in its off state allows extension, and it's unliganded, and when it's become liganded by SLEET or NEL2 or possibly other ligands, it becomes activated, which triggers a conformational change, recruitment of intracellular and or activation of intracellular effectors that signals for repulsion. Sorry. So there are 4 human robos. Drosophila has 3, and C. elegans has only 1, which basically allows somewhat simpler in vivo system to study SLEET robo signaling. The sequence of the C. elegans robo, which is called SAX3, is most similar to a well-known and well-studied cell adhesion molecule by the name of L1-CAM. And based on that, we think that roboreceptors are gene duplication products of an ancestral cell adhesion molecule. So outside, besides their role in axon guidance, SLEET and robo has many other functions in the CNS and outside the CNS, like in the organogenesis of kidney, heart, bone, angiogenesis, and so on. And consistent with that, deregulation in SLEET and robo are associated by a variety of developmental and chronic diseases, such as liver fibrosis, chronic kidney diseases, bone depletion, macular degeneration, and on. And most importantly, in cancer, where most cancer cases has deregulation of SLEET and robo. And by saying deregulation, I mean underexpression, reduced expression of SLEET due to promoter hypermethylation, and overexpression of robo. And a parallel overexpression in robo, one can imagine that robo being the brake and SLEET being the foot on the brake, that cancer cells avoid expressing SLEET, and as a consequence, there is an overexpression in robo, but it goes in vain because there's nothing to activate it. So one example for the therapeutic potential of SLEET and robo we can see very nicely in pancreatic cancer. So pancreatic cancer has many reasons why it's so difficult to treat, if at all. And one of the problems is very high genetic heterogeneity. And efforts made in recent years to map the genetic landscape of pancreatic cancer show that one of the genetic hallmarks of pancreatic cancer are deregulations in the robo and SLEET pathway. Here we see one example for the therapeutic potential of targeting ROBO by expressing ectopic expressingly slit in this pancreatic cancer xenograft model. We see that a result of the slit expression, vastly reduced primary tumor and elimination of metastasis. So clearly there is a lot of therapeutic potential within the slit and ROBO. However, there are to date, no ROBO-agonists or antagonists. And we thought that at least in part, the problem is incomplete understanding of the structure and mechanism of activation of ROBO. So to address this, Ruth Barak, a former graduate student in the lab, took it upon herself to express, purify, crystallize, collect data, and eventually determine the crystal structure of intact human ROBO2 ectodomain from the slit binding domain 1. It goes by color code from the slit binding domain 1 all the way like this to the membrane proximal D8. And it has this structure has a kind of how do you call it? Sika. Yeah? Hairpin, sorry. Kind of a hairpin structure with the yellow domain 4 right in the middle. Here are the transmembrane and intracellular segments, and they look like that because currently I have nothing intelligent to say about them. This structure was eventually solved using molecular replacement after many attempts using heavy atom soaks and selenomet derivatization failed. Because the crystal structure of the fragments that were available at the time that we addressed it were not useful in molecular replacement, Ruth started generating her own search model for molecular replacement, starting with the D7, D8, following D4, D5, and finally with D2, D3, that was the first brick in this domino cascade of structure determination that allows us to solve the structure. Later on, it doesn't work that well. Later on, Andrew McCarthy from EMBL Grenoble published the structure of ROBO1 domains 1 to 4. I'm telling you all this because this phasing strategy proved to be very useful, because especially when it comes to the structures of the D4 containing structure, because when we determine the structure of the D4, D5, we have noticed that there is a very interesting dimeric homotypic interaction in the crystal lattice of D4, D4. It was interesting because it was large, hydrophobic, and really conserved. It shows conservation, sequence conservation, even beyond bilateral creatures into Cnidaria, that is jellyfish. It is probably the most conserved molecular structure in ROBO. And we also experimented in solution, and we saw that it forms a dimer in solution with a dimerization constant of 16.9 micromolar. And one can imagine that with the confinement of the 2D membrane, this value would reach something like 1 micromolar or so, which is not very strong, but not very weak, and kind of ideal for targeting by monoclonal antibodies later on. This became exponentially more interesting when we saw and compared the structure of the EMBL Grenoble group of ROBO1, D1, D4, and we saw that it assumes exactly the same crystal packing through domain 4. Now, considering the difference in construct, in paralog, and in crystallization conditions and all, the chances for this kind of thing to be random are like zero, okay? So when we now look at this dimerization interface in the context of the full-length structure, of the ectodomain full-length structure, it is rendered here in white over a background of yellow D4. We see that this dimerization interface is actually blocked by D7, which immediately prompts the idea of auto-inhibited conformation, which is auto-inhibited for dimerization, something very similar to what people saw with EGF receptors, which are far more studied and characterized. So But this is kind of a model that one can work with, right? We can test in cells if this form is monomeric, and if removing these autoinhibitory constraints, it becomes dimeric and so on. The problem is that the intracellular segment of ROBO has no catalytic activity, so it's not easy to follow. For that, Julia Ghez, our lab manager, came up with this simple and tangible assay. She generated chimera between the ectodomain of ROBO and the intracellular segment of a receptor tyrosine kinase by the name of c-KIT. And the thing with receptor tyrosine kinases and c-KIT that by themselves, when they are unliganded, they are monomeric and non-phosphorylated, but upon ligand binding, it induces dimerization and phosphorylation of the kinase domains, something that is very easy to follow using anti-phosphotyrosine Western blot. So Julia generated the series of chimeras using the intracellular segment of the c-KIT with the extracellular portions of ROBO, and what she saw that the intact ectodomain of ROBO is dimerizing slightly, only a little bit, but when removing these autoinhibitory constraints, it dimerizes very well. And we saw the same thing also for ROBO1, for human ROBO1 and ROBO3, and for the C. elegans homolog SAX3, although to different extents. So one can say that ROBO3 is the least autoinhibited, and the SAX3 from C. elegans is the most autoinhibited. So to further test this activation model in which ROBO is monomeric and inactive, and upon assuming open conformation, it becomes dimeric and active, we wanted to have an in vivo model. So for that, we used the well-established C. elegans. Look at this. We use the very well-established C. elegans model for slit ROBO function. In this model, the track of the axon of the AVM neuron, in C. elegans, each neuron is tagged, named, and accounted for. So the track of the AVM neuron, this inverse L shape, is dictated by repulsive and attractive forces, by repulsive and attractive guidance cues. So the way it works is that slit is secreted from the dorsal muscles of the worm, and they respond on, they act on SAX3 or ROBO-expressing AVM, and by that, pushing it toward the ventral side of the worm. When either ROBO or slit or other components of this signaling system are missing, the axon assumes a direct anterior path, like you see here. So what we did, what Galit Yonath did, a former graduate student in the lab did, was on the background of these ROBO knockout worms, she introduced a rescue transgenes that carry either wild type or mutant ROBOs. And to cut a long story short, without getting into details, we see that when we remove the dimerizing D4, or even compromising D4 dimerization by point mutation, we see a complete loss of function. We see a complete loss of function, basically proving that D4 dimerization is necessary for ROBO function in vivo. When we remove the autoinhibitory domains, such as domain 7, what we see is that we get activity which is not dependent by slit. So it is as active even with the knockdown of slit expression. So what can we do? So this basically gives a functional in vivo corroboration to this model of monomeric closed conformation inactive ROBO versus open conformation dimeric active ROBO. So what can we do with this new information? One thing that we can do is basically generate monoclonal antibodies that will serve as agonists and antagonists. Antagonists by blocking the dimerization interface necessary for function, and agonists by blocking the autoinhibitory surfaces. So here we have example for one such antibody that we have produced. We basically screened through a human synthetic library for Fab fragments and isolated these Fabs that bind to Robo. And by doing differential binding of wild-type Robo versus point mutants in Robo, we could map the binding sites of these Fab binders. So here we see what we do with as a biological assay to test these Fab binders. We use the same assay that I showed you the movie before, only this one is with a much worse resolution. The movie wasn't ours, right? This one is. So, here in this assay, we also we used dorsal root ganglions, and we count the number of active growth cones. These are the dynamic structure at the leading edge of the axon, the hand-shaped-like structures, and before and after slit bath application. And we do the same with pre-treatment of one of these Fab binders that is mapped to block the D4 dimerization, and we see that it offers protection. Another thing that we do with this newly acquired information is to generate for the first time constitutively active mutants of roboreceptors by basically blocking or deleting or mutating the autoinhibitory surfaces. And here we generated a series of such mutants and packed them into lentiviral delivery system, and in collaboration with Eran Perlson in Tel Aviv University, tested these lentiviral clones on whole spinal cord neurons from mice. And what you can see here that activating mutations, constitutively active clones of both Robo1 and Robo2 results with elimination and retraction of the neurites that connect the various neurons. You can also see that it doesn't only eliminate the neurites, but also reduce the number of neurons. Basically, it also kills neurons. So sustained signaling of Robo leads to neuronal death. And we still don't know what is the pathway, what are the molecular pathways that basically induce this neuronal death. And this leads me to the second part of this talk regarding the SARM1 executioner for neuronal degeneration. So there are many reasons for neurons to die. Some of them are good reasons, such as in the course of development. It's perfectly normal and advisable for many neurons to die, but also bad reasons, such as in disease, neurodegenerative diseases, infectious diseases, and so on, and so on. And there are also as many molecular mechanisms that execute neuronal death following the various insults or signals that these neurons get. And there are also many inhibitors, so we know how to inhibit many of these different pathways, these apoptotic pathways in neurodegeneration. However, when it comes to axonal degeneration, there is much less information, or at least there wasn't much information, because genetic silencing of the various apoptotic and necroptotic pathways does very little to prevent axonal degeneration, for example, after severing. So in this course, we see, in this picture, we see an insult applied to an axon, and what follows is a very distinctive way of degeneration called Valerian degeneration. It involves granulation and elimination of this axon. And up until 2012, nothing was known about the apoptotic pathways that convey this phenomenon. And in 2012, in a Drosophila screen, it was found that a protein that blocking or eliminating a protein by SARM1 prevents entirely the process of this Valerian degeneration. And we now know, and a little bit later on, sorry, not now, a little bit later on, it was also found that mouse knockout mice for SARM1, which are perfectly normal in every respect. I mean, they live regular life in captivity. I don't know about wild, but in captivity they live a normal life with no sign of anything. They are basically resistant to Valerian degeneration, but not only for Valerian degeneration. I mean, these axons can last 2 weeks after severing, while wild-type axons degenerate within a day or two. So, but they're not only resistant to Valerian degeneration, they seem to be resistant for a whole variety of insults, such as other injury types, viral infections, Bunia virus, Zika, rabies, hypoxia. I think most importantly, they're resistant to chemotherapy-induced peripheral neuropathy. So something like 30% of people that receive chemotherapy suffer from injury to their peripheral nerves. And sometimes it's a lifelong condition. And these mice are completely resistant to it. So imagine a direct inhibitor for someone administered together or slightly before chemotherapy treatment. It might eliminate this problem. So what do we know about SARM1? What do we know about this protein? So it has 3 functional domains, ARM, SAM, and TIR domain. The TIR domain is the business end of this protein, and really surprisingly, it was found in 2015 that it carries an NADase activity. Up until then, TIR domains were considered, up until then and slightly after also, TIR domain were considered to only convey protein-protein interactions with no catalytic activity whatsoever. So these guys found that the TIR domain has a NADase activity which is comparable more or less to the other NADases we know. So it's not a weaker one. The same group also showed that the NADase activity of this TIR domain depends on dimerization, at least dimerization, also probably multimerization. So without being able to dimerize, the TIR domain by itself in low concentration carry no NADase activity. Only when it reaches a very high local concentration, the NADase activity occur. And they also found that what provides this dimerization ability are the SAM domains. The SAM domains provide dimerization, and the ARM domain provides autoinhibition. So without the SAM domain, without the ARM domain, the SAM and TIR domain construct is toxic to cells. And few hours after transformation, the transfection, the cells die. We were interested in studying the structure of this protein in order to better understand it and hopefully to develop treatment to block it in neurodegeneration. And Michael and Julia, Michael, a graduate student in the lab, and Julia, our lab manager, starting by addressing the crystal structure of the tandem SAM domains using X-ray crystallography, and we determined this structure that shows a closed octameric ring structure. Okay, it goes again by color code, the SAM1 and the SAM2, SAM1 and the SAM2, and both of them are engaged in parallel interactions on one plane that forms this closed octamer ring. We also showed that this construct is also octameric in solution, and more than that, we showed that the intact protein, the full-length protein, is also octameric in solution. We later examined this protein, this full-length protein, in negative stain EM, and we immediately got these beautiful micrographs and class averages that shows an inner ring superimposed perfectly by the crystal structure of the octamer with a peripheral ring with 2 different densities, presumably accounting for the TIR and ARM domain. Now at the age of the resolution revolution, we thought we wanted to take advantage of these new opportunities. Thank you very much, Professor Henderson, for helping humble biochemists in the Middle East for that. And the results exceeded our expectations. And we got very nice micrographs and class averages, and the density map is of a good quality, allowing us to dock and adjust the crystal structures that we have. So another group determined the tier domain here in red. And basically, the most conspicuous thing that comes to mind when looking at this structure is the striking similarity to other apoptotic complexes, namely the inflammasome and apoptosome. For some reason, these three apoptotic structures convey into this ring structures. I don't know what is the meaning of that. If there is a meaning at all, I'll just say that someone does modulate the inflammasome function in innate immunity, and there are reports that it directly interacts with it. So maybe there is some kind of structural complementation there. Consistent with the negative stain results, we see that the SEM domains form this inner barrel surrounded by the arm and tear ring. In mammals, there is also a mitochondrial localization tag, N-terminal localization tag in mammals, but not in other species. And here you see this view as if you are sitting on the mitochondria looking upwards. So this is a view from the bottom. This is a view from the top. And you can, in the next 3 slides, I'll analyze this structure one domain at a time, starting with the SEM domains. And for that, I'll remove some of these subunits. So it will be easier to see. Like that. Okay, so the SEM domains are the organizing center of this structure, and they're directly linked to the arm and tear domains. So the SEM1 directly linked through a well-ordered, very nicely visioned linker to the arm domain, and the SEM2 is linked via a linker that we don't see in the structure to the tear domain. The interactions that the SEM domains are packed side by side, and they are held like that due to complementing electrostatic charges that are very easily shown here, but also through hydrophobic inserts and grooves, complementing inserts and grooves in the SEM1 to SEM1 and in the SEM2 to SEM2. When we compromise these interactions using side-directed mutagenesis, and we test this using an NAD level sensor and cell viability count, we can see that when we compromise the SEM1 interactions, we see a loss of function compared with the constitutively active mutant that lacks the ARM domain altogether. The ARM domain is the autoinhibitory domain. So this is the most lethal form, and we see a loss of function when we compromise the SEM1 to SEM1 interactions. However, we don't see a very impressive inhibition when we mutate the SEM2. And so why would we need the SEM2 at all? So we think that the purpose that SEM2 serves is to allow this ring allow this oligomer to assume a ring structure by confining it to one plane. In other cases, when SEM domains mediate multimerization and there is only one SEM domain, what you get, in several cases actually, what you get is an open-end helix because it lacks the geometrical constraints that will keep it in one plane. So we think that this is what the SEM2 is good for. The ARM domain is the only part of the structure that we have built from scratch, that we didn't have a crystal structure template for. And they're arranged like roof tiles, one covering the next. And each one originates from one SEM domain, but basically mostly interact with the neighboring SEMs. Altogether, there aren't so many strongly looking interactions in this packing, not between the arm and the neighboring arms, and not between the arm and the SEMs. The arm domain, it's short for armadillo because of its resemblance to the creature, armadillo. It has a concave face and a convex face on which the tear domain docks. Each tear domain docks on, actually interacts with two neighboring arms, with one a major interaction and with the second a minor interaction. Probably the most interesting thing to see here is that the NAD binding site of the tear domain looks completely accessible in this structure. So we think that the autoinhibition that the arm domain serves in this context is not by blocking, is not by interfering with NAD binding, rather by the very docking itself, because in this way the tear domains are separated from each other, unable to form the dimers needed for NAD catalysis. When we look closer at the major interaction site between the arm and the tear domain, and here it's a like open book representation of this interaction and a mapping of the interacting site, one thing that it's nice to see is the sequence conservation of these surfaces. Thank you very much, Neil, for that, which highlights the importance of this interaction. When we compromise this interaction by point mutations in the arm domain, we see that what we get is a constitutively active form of SARM1 similar to the elimination of the entire arm domain itself. And we think that this interaction is the key for SARM1 structure. By maintaining this interaction, when SARM1 maintains this interaction, it's inactive, it's autoinhibited, and when it loses this interaction, when this interaction breaks, it assumes the active apoptotic form of this protein. So here is a model, and this is my humble representation of mitochondria. And I'll leave you at that in terms of our results, but I will just say that probably the most interesting aspect of this work is not addressed yet. And this is what exactly activates SARM1. What drives this structural transition from this autoinhibited compact structure to what is necessarily an open and dimeric structure of the tear domain. Is it oxidative stress, and if so, how? Is it metabolic stress? Is it binding of another protein or another factor, another small molecule, as was suggested before? Or rather, is it the losing of a trophic factor? Maybe a trophic factor, I tend to think so, that under stress condition, there is a depletion of a certain trophic factor that breaks this compact conformation. I'll end by thanking my fabulous lab members, students who did the work, my great collaborators all along the way, and special thanks to Ran Zalk, who is in the audience, who runs a top-notch facility, the best facility that I know. Without them, nothing of this would happen. The synchrotron beamline scientists, with all their advice and help and patience, cryo-electron microscopes. Now we use the ESRF facility for that, and the funding agencies, and you for listening. Thank you very much.
Okay, questions?
Maybe I missed this, but did you actually find that the SARM1 binds to mitochondria?
Well, not we, but others showed that mammalian SARM1 is found in cells in cytosol and mitochondria.
Okay, so the octameric structure might be important to bind to some similar structure in the mitochondrial outer membrane?
Well, it's a very interesting question, because actually there is well, not in the context of mitochondria, but there is an extracellular protein by the name of RS1 that is the only other structure that looks like that. It is an octamer, okay, only other structure that we know of. And it is an extracellular protein that organizes sodium-potassium ATPases, transmembrane sodium-potassium ATPases, and it was reported that SARM1 interacts with that. So one can, in a dream experiment, would reconstruct this RS1 extracellular ring octamer organizing 8 sodium-potassium ATPases and intracellular SARM1. Yeah.
Do you have any connection with apoptotic conditions?
Connections how?
In the structure or function, something like this.
Well, it induces apoptosis, yes. I mean,
So it may be that cytochrome C may interact with this. Did you try?
I think it's a standalone. It's a separate line of, it's a separate pathway of apoptosis.
Yeah.
It is a standalone. It means only that. It reduces dramatically the amount of NAD in the cell below a certain threshold.
Okay.